Hey guys, today we're going to start talking about topic 2.6, the structure of DNA and RNA. Now remember, DNA is the molecule of inheritance, and it determines the traits of an organism. It's found in the nucleus of eukaryotes and the nucleoid of prokaryotes. So remember, eukaryotes have a nucleus and prokaryotes don't. DNA is housed inside of the nucleus in eukaryotes, and that's where it stays. It doesn't ever leave. It's protected in that nucleus. Prokaryotes are different. They're more simple. They're simplistic. They're bacteria. Their DNA floats around inside the cytoplasm in an area of the prokaryote called the nucleoid. Okay, but there's free access. It's not contained within its own nucleus. So just as a refresher, you can think of your DNA as like a blueprint to build a boat, right? Your DNA is the instruction manual on how to build all of the chemical components that make you. So, all the proteins. So, DNA contains the instructions on how to build proteins. Now, DNA does not make proteins. DNA is just the instruction booklet. So, if you buy a couch from IKEA and you get it home and you open it up and there's no instruction manual in there, right? You're in trouble because you don't know how to put it together. You can think of your DNA as like that instruction manual. It contains the information on how to build proteins. Remember, proteins are built from amino acids. So there's two major types of nucleic acids that we're going to talk about, DNA and RNA. And they are different, and you do have to know the differences between them. However, both are composed of smaller units called nucleotides. Now let's go all the way back to last semester when we talked about macromolecules. We talked about the monomers, which are the simple building blocks, and how you put those together to create polymers. Well, the last set of macromolecules that we never talked about were nucleic acids. I waited till now to talk about nucleic acids because we're going to start talking about DNA replication and transcription and translation. And that's when you really need to know about nucleic acids. So the monomer, or the simple building block of a nucleic acid, is called a nucleotide. Nucleic acid, nucleotide. You put multiple nucleotides together to create a polymer, and that polymer is nucleic acid, either DNA or RNA. So understanding number one, the nucleic acids DNA and RNA are polymers of nucleotides. So a nucleotide, just one nucleotide, has three parts. So if I go back, each one of these circles that represents a nucleotide, each one of those circles has three parts. It looks like this, or more simply, like this. We have a 5-ringed sugar. You should immediately be able to identify that as a sugar or a carbohydrate because it's got that funky ring structure. Now, this is a 5-ringed sugar. This is ribose. RNA, ribonucleic acid, right? Ribonucleic acid has ribose. Ribose. You have this circle over here that's a phosphate group, and then you have a nitrogenous base. Now, there's different kinds of nitrogenous bases, and we'll talk about that in a minute. But for now, it's important that you understand that when we draw nucleotides, typically we draw them with a circle representing the phosphate, a ring for the ribose, and some sort of shape, a rectangle. Sometimes it's a rectangle with a triangle cut out for the nucleotide. Okay? So you'll hear me use the word nitrogenous base and just base interchangeably. And there's different kinds of bases. We'll talk about that in a second. In your review questions, they do ask you to draw a simplified nucleotide, and this is what they're looking for. They want you to label it, but like they're looking for the circle, the pentagon, and the base. Now we put nucleotides together to form DNA and RNA. You can see there's 1, 2, 3, 4 nucleotides here. I did that by looking at the number of phosphates, the number of ribose molecules, and the number of bases. And here you can see an example of 4 different bases: cytosine, guanine, adenine, and thymine. So if I had to draw circles around each of the bases, if I had to draw circles around each of the nucleotides, right? Here's 1, here's 2, here's 3, and here's 4. Now, just like all the other macromolecules that we talked about, you link monomers together to form polymers using dehydration synthesis. And we'll show you that in a second. The bond that links nucleic acids together is called a phosphodiester bond. And that makes sense because it's a bond between the phosphate, right? And di means 2, right? Ester is something to do with organic chemistry that we don't need to get into. But the point of this is you should be able to tell that the phosphodiester bond is between the phosphate, right, which is a circle, and the ribose below it. So we have a phosphodiester bond here, we have one here, we have one here, right? And these are linking 2 different nucleic acids together. Now, you might be saying, well, what about the other ones? Aren't those phosphodiester bonds too? These ones? Because they're linking a phosphate and a ribose. Yeah, but they're not linking nucleotides together because remember, this is one nucleotide, right? Phosphodiester bond 2 different nucleotides. So the bond here is linking this nucleotide with this nucleotide. The bond right here is linking this nucleotide with this nucleotide. And the bond here is linking nucleotide 3 with nucleotide 4. Okay, so phosphodiester bonds are the bonds that link nucleotides together. And they do it along the outside of the strand, and we call this the backbone. This is the backbone of the DNA. When you think of DNA normally, you probably think of like the double helix, right? Well, that double helix, if we stretch it out into 2 straight lines, looks like this. This is one straight line. So the phosphodiester bonds that we're looking at are the links between nucleotides on one strand. And this will make a little more sense as we go on. What's important is that you can identify where the phosphodiester bonds are, and they're between the ribose and the phosphates of nucleotides on the same strand. So here we can watch 2 nucleotides joining together through a condensation reaction. We have a nucleotide here, we've got the phosphate group, we've got the ribose, and we've got our nitrogen base. Down here, we have another nucleotide. We have our phosphate group, we have our ribose. I don't know why that one's not red. And we have our nitrogen base, right? Now, if you look at our phosphates, I have a little hydroxyl ion that's noted right here, right? And I also have the hydrogen noted here that's sticking off this sugar, right? Now, condensation reactions, that means water falls out, right? So just like all those condensation reactions we talked about last semester, we're going to link these 2 by pulling out the hydrogen from here and the oxygen. This oxygen will then be bonded to this part of the phosphate. So let's watch. And remember, this is dehydration synthesis. Oh, now it's red. So you can see this is that phosphodiester bond right here between the ribose and the phosphate. Now, similarly, when we want to break them apart, we just add that water back in, right? and that's hydrolysis. This is a review from last semester. Here comes our water molecule, and we split apart those nucleotides. So in summary, this is a diagram. It's a little complicated, but it's just showing you the same thing. We have a nucleotide here, and we have a nucleotide here. We have our hydrogen, hydrogen, our hydroxyl right here, and we have a hydroxyl right here. We're going to take this hydrogen, and this oxygen and remove it. And that way, this oxygen can bond with the phosphate here, and we're going to create that link. And that's a phosphodiester linkage. So I said before, there are 5 different nitrogenous bases. So these are the third part. So we have our phosphate, we have our ribose, right? And then we had that nitrogen base. So this is the part that we're talking about. There's 5 different kinds. There's adenine, guanine, cytosine, uracil, and thymine. Now, not all of these are found in both DNA and RNA. Uracil is found in RNA only. Thymine is found in DNA only. Cytosine, guanine, and adenine are found in both. So if you're looking at a molecule and you see T's in there, you know it has to be DNA. If you're looking at a molecule and you see U's in there, you know it's got to be RNA. Now, you don't have to know the specific structure of the different bases. You just need to know that there are 5 different types and that thymine is only in DNA, uracil is only in RNA, and the other 3 are in both. So let's compare and contrast a little bit. Understanding number 2 says DNA differs from RNA in the number of strands present and the base composition and the type of pentose. So this is really a summary of the differences between DNA and RNA. So RNA is a single strand. Don't worry, nucleobases are the same thing as nitrogenous base. Same thing. Nucleobase, nitrogenous base. So RNA, single-stranded, right? That's number 1. Whereas DNA is double-stranded. In DNA, the sugar part of the nucleotide is deoxyribose. So you might be saying, well, what the crap does that mean, deoxyribose? Well, remember, we're talking about sugars, and the formula for ribose, I believe, is, let me look it up. I'm pretty sure it's just standard. It's 6-12-6, but I'm just going to make sure. Ribose formula. C5. Okay, so ribose we have C5, and it's a carbohydrate. So we have that 1-to-1 ratio, right? So C5H10O5, right? Deoxyribose. Deoxy. D means not, oxy means oxygen. So we're looking at a ribose that's missing an oxygen. So the chemical formula for deoxyribose all you do is just remove one of those oxygens. So it's C5H10O4. Okay, so that's another difference between DNA and RNA. And remember, we're talking about the hexagon, the sugar, and the phosphate. I'm sorry, the sugar in the nucleotide. Yeah, sugar in the nucleotide. Okay. Another big difference that we just talked about, DNA has T, but no U, and RNA is the opposite. RNA has U, but no T. Okay? All right, so that was understanding number 1 and understanding number 2. This is important. I have seen exam questions that ask you to compare and contrast DNA and RNA. Understanding number 3. DNA is a double helix made of 2 anti-parallel strands of nucleotides linked by hydrogen bonding between complementary base pairs. So, like we said, DNA is made of 2 strands of nucleotides that fold into a double helix structure. The strands are anti-parallel, which means they're oriented in opposite directions. Okay, so what does that mean? So if we look at the picture all the way on the left, right, you can see that all the, this is deoxyribose, right? So you can see all these ribose molecules are pointed up. DNA has rules. On the other side, they're pointed down. That's what anti-parallel means, right? You can think of it as like, just like on a road, right? Like when you're driving, you always drive on the right side of the street. And it's just a rule that everybody follows so that you don't get into accidents, right? Well, DNA has rules too. It has rules just like that, sides of the street, and they're opposite of each other. Now, there's something called prime, 3 prime, and 5 prime. And those are to indicate the directions in DNA. So let's see. I'm going to label this right now so that you can tell which way is the 5 prime and which way is the 3 prime. Okay, so when the hexagons point up, they point towards the 5 prime, right? They come from the 3 prime. Now, same thing on the other side. The arrows point to the 5 prime, right? The sugars, the riboses, the deoxyriboses, are going to point towards the 5. They're always pointing towards 5. And the other end is 3. Now, you might ask me, like, why is it 5 prime and 3 prime? And it has to do with the orientation of the hydroxyls coming off of the ribose. And you don't really need to know that. You just need to know that the strands are anti-parallel, right? And you can tell which is the 5 prime and then the 3 prime by which way the riboses are pointing. Now, this will become important when we talk about DNA synthesis because that can only happen in certain directions, right? So from 5 prime to 3 prime. So you can think of this as directions to and from, okay? Now, we said earlier, it's the phosphate groups and the sugars that make up the backbone. Now, I said earlier, I called it the backbone. So the blue part, that's the backbone. And that's the sugar and the phosphate, right? And the red part is the backbone. Now, the rungs, if this was a ladder, you made it straight and it looks like a ladder. The rungs, this part, these are the bases. So the bases are the steps in the ladder, and the backbone is the phosphate and the sugar. Now you also notice that the bonds between the base pairs, so now let's look at, I'm going to pick a different color so that we can see. Okay. So now if we're looking at the bases, right? We're looking at the bases. You'll notice that between C and G, there's 3 hydrogen bonds. Between A and T, there's 2. And that's on purpose. This bond, or these bonds, are hydrogen bonds. So there's 2 different types of bonds in DNA. There's the phosphodiester bonds, right? Actually, be that one. Diester bonds. And then we have the bonds in between the bases. Those are hydrogen bonds, okay? Make sure you're drawing lots of good diagrams in your notes because if you can draw a diagram that has all this stuff, it'll help you answer questions. So now we're going to talk about DNA base pairing. And these are also called I think it's Chargraph's rules. I might have spelled his name wrong. Might be Chargraph. But these are the rules that say C goes with G and A goes with T. Now, two sides of the chain are held together by hydrogen bonds. We just talked about that, right? So these are H bonds. Now, there's some math that you can do to help figure out base pairs and how many there are. So